Hello, here we are in the lab of our school, and we are going to show you how PCR and electrophoresis can be used in forensic analysis as a part of the process of DNA profiling. As you can see, we wear lab coats and gloves to prevent contamination of the biological samples. We are going to show you a murder case in which DNA samples involved in a crime must be compared to find possible matches between the victim and the suspect. The first part of the experience is PCR, which exponentially increases the copies of a sequence of DNA. All the DNA samples involved in the case will undergo PCR, producing a large amount of copies of the original sequence. The second part of the experience, electrophoresis, will isolate the same sequences by size and allow us to measure them and find possible matches.
In PCR for DNA fingerprinting, only small parts of DNA are amplified. These sequences are different in each person. In fact, we all share 99.5% of the human genome, and only in the remaining 0.5% there are some sequences, called loci, that differ from individual to individual in a limited number of variations. These variations are called alleles. As a result, we now analyze the same locus from different DNA samples, and we will obtain different allele values. The DNA of each person has only 2 alleles for each locus, one from each parent. In this way, the 2 allele values we get from each sample will help us compare the DNA and find any possible matches.
To carry out the procedure of PCR, in each workstation we need a refrigerator rack containing empty PCR microtest tubes with caps, another refrigerator rack containing test tubes in different colors with different DNA samples from the crime scene and from the suspect, A, B, C, and D. We also need the PCR microtest tubes adapter because microtest tubes are too small to be directly inserted into the centrifuge and in the thermal cycle. Another test tube contains the master mix with all the substances needed for PCR application, a large amount of nucleotides, 2 types of primers, heat-resistant polymerase, buffer, and so on, everything except the original samples to be amplified. To deliver the right quantity of substance into the PCR microtubes, we need the micropipette, a precision tool which will help us to deliver the right quantity of DNA and of the master mix.
On this whiteboard, there is a scheme of what we are going to do. In the 5 empty labeled PCR microtubes, we introduce the corresponding DNA samples. Then, in each PCR microtube, we add the master mix. Now we move to the workstation to carry out the procedure. We label the empty PCR microtubes with the initials of the corresponding DNA samples to be added in each. We turn on the Bunsen burner to create a sterile environment. In each PCR microtube, we add 20 microliters of the corresponding DNA samples. We use the micropipette, changing the pipette tips anytime to avoid contamination. Then, in each PCR tube, we add 20 microliters of the master mix. Here again, we change the pipette tip every time. Now we close the 5 PCR tubes, place them in the adapters, and put them in the centrifuge to mix the components.
After the centrifuge, we put the 5 PCR tubes in the thermal cycle. This instrument is able to increase and decrease the temperature for the required amount of time to start the process of polymerization. It can carry out the amplification of many DNA samples at the same time. When the process is over, products are removed from the thermal cycle and are ready to undergo electrophoresis. The process of electrophoresis isolates the fragments and measures them by size. This electrophoretic chamber is the instrument used for this purpose. As you can see, it has 2 wire electrodes at each end. Near the negative pole, there are some wells created on the agar gel, where we put PCR products. each DNA sample in a different well. In the first well, we also put an allele ladder, a size standard containing all the possible allele values for that locus. In this way, we can have reference values to measure our DNA samples.
When a direct current is passed between the electrodes, DNA fragments, which are negatively charged, will move toward the positive pole, while the gel works as a molecular matrix. The shorter fragments will move faster, the longer will move more slowly. Over a period of time, the fragments of the same length will move together at the same speed and form single bands in the gel.
After staining and destaining the gel, we will see two different bands of DNA in each lane. Each band represents an allele for a specific DNA locus. By making a comparison between the bands and the allele ladder, we can determine if the test has given reliable results. At this point, we can find out if there is an exact match between the crime scene DNA and one of the suspects.